Good afternoon, everyone. We will continue with our work on, or our lecture on lambda phage, lytic and lysogenic decision making. And we are learning in terms of gene regulation, not necessarily as a whole virology part of trying to understand lambda phage life cycle and so on. So I will limit this lecture only about understanding the regulation, right? We learned about transcriptional regulation. In that, we learned about lacoperon, tripoperon, NTRC, and many other, some other arabinosoperon, and so on. So in that light, we'll also discuss about lambda phage. So in the last class, I already gave you an introduction. Lambda phage is a temperate phage, which means that it can undergo either lytic growth, where it will just infect a bacterium and parasitize, in the sense that it will use up all the resources that are present in the bacterium, and it will lyse the cell, and new phages are released. So all reproduction happens, bacteria is dead, the host is dead. That's why unlysed. So you call it as lytic growth. The other type is called as lysogenic growth, where it is kind of two DNA molecules, if I may use anthropomorphism, as if DNA molecules have some consciousness. Just think for sake of understanding. It's like you have bacterial DNA here, and there is phage DNA. It's as if there is a war between these two, and they both decide to kind of make a kind of truce where they both survive, right? The survival of phage is dependent entirely on the bacterium. And the bacterium, if it allows the growth of phage, then the bacterium is going to die, as you see it in the lytic growth. So this is kind of truce between two DNA molecules. One is bacterial genome, the other one is phage genome, and they kind of become one, and they exist. I mean, if you see, eventually the bacteria will lose. But we are going to just discuss about whether, because when I say lysogenic, at some point, the phage might get induced. This is prophage, right? When it is integrated in lambda phages, or viral genome is integrated into the host genome, you call that part of the genome as prophage. So prophage might excise, as we learned in site-specific recombination, that integration may happen, excision may also happen. And excision, then it means it is induction, and it is going to take lytic cycle. But otherwise, it is mutually beneficial to be in lysogenic cycle, in the sense that bacterial genomes are growing, as well as phage genome is also getting amplified, or propagation is happening. So whether lytic cycle should happen or lysogenic should happen, how is it regulated or decided, is what is the essence of this particular course or lecture. So we should understand the lambda phage genome itself, the organization. And lambda phage has a capsid, that is the proteinaceous coat. It also has a stalk or tail and some things like this. So inside which you will have the genome, which is usually linear when it is in the phage. In the capsid, the outer coating, proteinaceous coating, is called as the capsid. So if one phage has to propagate, that means it has to form more capsids, genomes, and so on, right? And that is meant by propagation. So it needs, these are the head proteins that synthesize the capsid. The tails are these that produce the other accessories, which are essential or important for interacting with the bacterial host and then injecting the DNA, right? So for that, tail genes are also important. And there are lysis proteins, the proteins that will allow, when produced, will lyse the host cell membrane. So bacterial cell, once it is made, and you have, say for example, it's undergoing lytic cycle, and there are a lot of phages that are made and new ones, and they should be released out. And for that release to happen, and please bear in mind that a phage as such has no value or life activities. If you take a virus outside, if you see it, it is almost as good as non-living things. No stimulus to non-response to stimuli, no movement, nothing. It only flows along with the water, air, droplets, and so on. Right? Virus outside the host is considered non-living. Virus inside the host is considered living. So viruses are between living and non-living things. That's how people view it. So these phage particles should be released out, which means the host membrane should be lysed. And these are the proteins that are, these are the genes that produce proteins for lysis of the host genome. And here are the phage DNA, as I said, should also be made. The phage DNA, multiple copies should be there. Empty heads is useless, isn't it? So if you look at it, all the genes that are responsible for making more copies are on this side, right? On our right side. If you see the other side, you will see that there are proteins for recombination. You will see proteins for, such as integrase and excisionase, which are important for integration of the phage genome into the bacterial genome. So if you see on this side, these are more into making lysogeny. And on the other side, it's the right side, or I'll put this also, the left side is more into recombination and formation of prophage, in the sense that integration of phage genome into the bacterial genome. On the right side, if you see the clockwise, you will see more of the genes to make more phage particles and lysis of the host. So these are genes are more into lysis of the host genome, right? So these are, there are many other things that are indicated, and I think we will have to understand it. So I will just discuss those again as well. So there are three important proteins or genes: C1, and that's Roman numbers; C3, where is C2? Here is C2. We'll get them. So there are three important proteins that are important for lysogeny. Lysogeny would, those that are promote lysogeny are C1, C2, and C3. Okay. Those that promote lytic life cycle is CRO. So what in the rest of the courses, what in the rest of the class, what we will be discussing is which proteins are expressed higher in concentration, which ones are not. And so the first thing is we should know the functions of these genes, and the genetic elements. Genetic organization or architecture is what I should say. But yeah, I hope that is what we will discuss: genetic organization and the function of these proteins and the consequences of those proteins. So PL, L is all for left, R is for right. If this is what gene expression happens in this direction, then it is mostly favoring the lytic life cycle. If it is going the other way, then it's mostly favoring the lysogenic. But we will get into details now. There are about 50 genes. I think it's about 48 kb or so, say approximately 50 kilo base pairs. And as I said, it is usually in linear when it is in phage, but once it is integrated into the host cell, it becomes circular, right? and that is done by cos sites. So typically, outside the cell, it is something like this, which has a small overhang like this. Here you see there is a overhang, and these bases are sticky ends, or in the sense they are complementary, and these are called as cos sites. If you are ever going to learn about recombinant DNA or genetic engineering, that is an elective for you when you come to 7th semester. There we discuss, I take that course as well. So in cos sites are important for that, for several applications. So once the DNA is integrated into the cell, it becomes circularized and sealed by the ligase. The same ligase we know it in during DNA replication that we learned. Okay, now let's look at close look at this area. This is the area that decides. This is the area of the genome that decides whether it's going to go lytic or lysogenic. So let's see the organization C1, and this is Chrome. Those are important things. C1 is for clear plaques, in the sense if there is a mutation in it, it forms clear plaques. Clear plaques indicates lytic. Okay, I will try to have a picture of it in the next time. You will see that. So PL is P is for promoter towards the left. PR is for the promoter for the right. And there is another promoter called as PRM. RM is for repressor maintenance, like it's given in here. It is a weak promoter, and upon transcription initiation from here, you will have the production of C1 protein. C1 protein is a repressor. We will discuss that. And CRO is also another protein that binds to DNA. So it is also it's controller of repressor. So typically in lytic phase, you will see that PL and PR, they both are switched on, while as in the lytic cycle, you'll see PL and PR are on. So you'll have the production of CRO proteins, which will also lead to the repression from PRM, and therefore it will undergo lytic cycle. The genes responsible for head proteins, tail, lysis, and all, they will be expressed. In lysogenic life cycle, what happens is that PL and PR are usually not expressed, whereas PRM is expressed. PRM is for repressor maintenance. What is that repressor that we are talking about? That is C1. C1 is a repressor and this promoter allows continued production of C1, although at a lower level, and because of which we call it as, or it was named as, promoter for repressor maintenance. So now we'll go even closer and try to look at what's happening here. So this is C1, and here is CRO. If you see the intergenic region, when I say intergenic, that is the region between these two genes. There are several operators, O for operator, and because it's on the right side, we'll say OR1, OR2, and OR3. But the promoter region PRM is covering, or OR3, operator R3 is present within PRM, promoter for repressor maintenance. And OR1 and 2 are kind of within the promoter for PR. So you'll see that these are the regions, how they are, -10 and -35 and so on. So we will come to that. Before, let's have a look at lambda repressor. Lambda repressor produces a protein that has kind of two domains. One of it is a DNA binding domain, and the other one is for dimerization and tetramerization. Okay, I hope you understand that. Dimerization means two proteins will come together. Tetramerization is 4 proteins coming together. And this activation domain is about binding of RNA polymerase and other things, activation, transcriptional activation. Okay, I hope you will be able to remember that. These three, OR1, OR2, and OR3, share certain degree of similarity, and they are overlapping with PRM, promoter RM, and promoter right. So the what lambda repressor or CRO does is something interesting in a way, in the sense that if C1 is produced, C1 produces a a protein with these domains, and they have higher affinity for OR1 and then OR2. So if C1 protein or lambda repressor, you call it as, binds to OR1 or OR2, CRO protein will not get expressed. Why is that? Because PR is occupied by the repressors here. Therefore, transcription will not happen and CRO cannot be produced. In that, if CRO is not produced, lytic cycle does not happen, and it will allow promotion of lysogenic life cycle. But if CRO is first produced or transcribed, if CRO is first produced, CRO protein binds as a dimer to OR3 preferably, and that will prevent the production of C1 because the operator R3 is within promoter repressor maintenance, PRM. Therefore, you will not have C1 expression or transcription. So if C1 is not produced, it is going to undergo lytic cycle. Okay, so the key here at this juncture, at least the way we should remember, is what proteins are occupying these regions. If lambda repressor is produced, CRO is inhibited, C1 is, or lambda repressor is produced more. If CRO is produced first, then we will have the production of CRO a little bit, but it will be repression of C1, lambda repressor, because of which it will undergo lytic cycle. So there are three important proteins that I discussed about having for the lysogeny and lytic cycle, or four in total, I said. 3 for lysogeny, that is C1, C2, and C3. Now we are trying to expand and get in more details. Until the last slide, we only discussed from here to here, C1 and CRO, right? So these are the four genes whose products are important in the decision making of whether it should go lytic or lysogenic. And the key is dependent on C2. We will see in the following slides why that is. But otherwise, I would want you to remember this PR we have already discussed. And here we'll have OR3, LGS3, and somewhere 2, and OR1, right? So when when PR is activated, or that promoter is active, it will have the transcription, say, we'll put it this way, and it will produce the CRO protein as well as C2 protein. And there is another protein that is called, another promoter that is called as PRE, E for establishment, repressor establishment. What is this? PRM is for repressor maintenance. So for the production of C1, the production of C1 is dependent on two promoters. One of it is PRM. We discussed and we told that we spoke that it is actually not so strong, or it is actually weak. I'll put it upwards. PRM, and there is another promoter from which C1 can be produced, that is PRE. This is the first promoter that will, when we say, when is PRM much more active? When you already have lambda repressors bound here. That is when they can activate the transcription from PRM. And where did that lambda repressor come in the first place? That comes from PRE, that is repressor establishment. It will allow the transcription of PRE will allow the transcription of C1. So this is transcription happens, and from there you will have the lambda repressor produced. So here is CRO, but CRO ORF will not be in this mRNA because they are encoded on different strands. They are not on the same strand, right? So try to remember them like that. So I hope you understood. Lambda repressor can be transcribed from two different promoters. The first one, most active one, is PRE, repressor establishment. Once that happens and C1 is produced, then it will allow the production of more C1 through activation of PRM, repressor maintenance. Yes. So now we are going to, when I said about PR, I said it will produce both CRO as well as C2. Okay. Now I think we are in the actual complexity. It's a lot of complexity. It's all right. See how much you understand. I don't think I will ask this in the exam, but it's a good way to learn complex things. So C1, CRO, and C2. Say, as soon as a phage infects the bacterium, you'll have certain promoters active, something like, say, PR is active, and then it is going to produce CRO as well as C2. C2 has a different functions. Typically, if you have higher concentration of CRO, if you have high concentration of CRO, it will favor lytic cycle. There are many other. First thing is CRO will repress the production of lambda repressor, and when that doesn't happen, then you will have the production of genes that are responsible for lytic cycle. But there is C2 is something that is another one that plays a role. C2 binds to a site. It is also producing a protein. It is also a protein, DNA binding protein. It's a regulatory protein, and it binds to a C2 binding site that is just nearby the C1 operon. Okay, C2 is produced, it binds here. Once it binds here, it acts as an activator. Activator for PRE, repressor establishment, because of which what will happen? A long mRNA is produced, and in that, C1 is also expressed. Once C1 is expressed a little bit, it will go and bind to the OR1 and OR2, and it will repress CRO protein, and it will activate the production of more lambda repressors from C1 gene. Okay. There is a problem, however, is that the C2 is not very stable. So although it is produced, it is also highly prone to degradation. So if C2 is degraded faster, okay, then you will not have this step. That means it will not go and bind to the C2 binding site. If C2 binding site is not bound, C2 does not bind there, PRE is not activated. I'm trying to go in the reverse way, okay? So try to understand it this way. As soon as infection is there, PR is expressed, CRO and C2 proteins are produced. Typically, C2 is not very strong or not very stable. So it might get degraded by the cellular proteases or host proteases. If it is degraded, you will not have enough concentration to bind to the C2 binding site. If C2 binding site is not occupied by C2 protein, then it does not activate the PRE, P promoter of repressor establishment. If PRE is not, the promoter is not activated, there is no production of lambda repressor. If lambda repressor is not produced, you will not have repression from PR, right? And you will also not have activation from PRM, so you will not have any production of lambda repressor. And then in that, which would also mean that you will have more production of crow, and eventually leading to lytic cycle. Okay, so these are the complex network of events, and the cell and the phage has events happen in such a way that will favor the phage propagation. Okay, I don't want to say that phage is smart and phage can decide whether it has to undergo lytic cycle or lysis cycle. That is anthropomorphism, saying that phage has something like conscious and it has a directed one, and it is doing events based on, or it's responding to stimuli, like that kind of things. It's not proper biological explanation or scientific explanation like that. Instead, we'll talk about events, which one will happen and which one will not happen. Okay, here is the situation, and we need to bring in, I think in the next slide, we will get to that. So when is lytic cycle favorable? When is lysogenic favorable? I think I should have used an empty slide, but I'll try to use this space. Say, for example, there is a tube and there is one bacterial cell. And I will also put this way. There is a tube and there are, say, 5 bacterial cells, or say 10 of them. Okay. In this case, if infection has happened, say, for example, these are bacterial cells that I have drawn, and I'm going to put some phages in there, and I'll use a different color. So I'm going to use a phage. Sorry. I will use these as phages. I'm just putting a small circle. Say there are 5, or yeah, say 5. Here you have also 5 of them. In which case is lytic cycle better? In which case is lysogenic cycle better? Although the numbers that I have chosen are actually very, very small numbers, but it will still give you an idea about what I'm trying to say. Here, if one phage infects it and undergoes lytic cycle, say from 1, because of which it will form 10 more new phages. That number is also very less, actually. It can form hundreds or thousands. But I will just put it like this. One phage has infected the only bacterium that is available, and it has produced total more of 10 more phages. So how many do we have now? We are left with 5 and 10. We have about 15 phages, new phages that have formed. In this case, 1, 2, 3, 4, 5. And say I have 6, right? We have taken 6 here, so I'm also taking 6 here. Now, each one of them has infected one bacterial cell. And then there are, say, in this case, 1, 2, 3, 4, 5, 6. So you'll have more 60 additional phages produced. 6 are already consumed, right? 60 new ones are produced. So this is assuming in both the cases there is lytic cycle. And then what will happen to these phages afterwards? Is it better to be like that? I mean, when it is outside the cell, usually the only way is to go downgrade or degrading, right? If it's not good. Let's see a different scenario. Assume that in situation A, and this is situation B, we'll talk it like that. What will happen if lytic cycle has happened in both the cases? Sorry, lysogenic has happened in both the cases. There is, in this case, if lysogeny happened, what is there? It will form a prophage within the host cell. In this case, it will form a prophage in the host cell. And you will have 6 genomes, 6 prophages total. And here you will have 1 prophage, right? The bacterial cell can divide further based on the number of nutrients available. Let me erase this and try again. If the cell numbers are very less, what is better? Is it better to undergo lytic or lysogenic? That is the question of it. So if there is only 1 bacterial cell, and if you infect and release 10 and they all die, there is no point of it. And if you there is I will get to this slide, and then we will also discuss the same thing. What is better in the based on the context? There is something called as multiplicity of infection. It is like I said, we have 1 bacterium, and say we have 5 phages. How many are infecting that bacterium is called as multiplicity. If only 1 bacterium, 1 phage infects 1 bacterium, then you can say MOI is 1. If there are 2 or 3 infecting 1 bacterial cell, 3 phages infecting 1 bacterial cell, then MOI is 3. Okay? Multiplicity of infection. If the bacterial cells are in the count of bacterial cells are more than that of phage, okay, phage numbers, typically MOI will be 1 or lesser because bacteria are more. But if bacterial cells are lesser, multiplicity of infection could be higher. There will be 2 or 3 phages infecting 1 bacterial cell. Okay? And that condition is not a good, which means that the whole number of host cells available outside are not in great number. That's not good for the phage, in a sense, and for propagation of the phage. Sorry. So, if there are very high numbers, I will just want to do to talk about what will happen if MOI is more. I'm saying MOI is, say, 3. And I will compare it with a condition where MOI is 1. Okay? So in this case, how much I want you to focus on C2. What are the properties of C2? It is produced along with chrome, and it is very unstable protein. Why is it unstable? Because of a protease. A protease is there that degrades the C2 protein. So C2 protein is important for binding to C2 binding site, and that is important for transcription from PRE, repressor establishment. That is only then you will have lambda repressor transcription or expression. So what will you expect in this case? If there is only MOI of 1, then you will have very little C2. Why is that? Because there is only 1 C2 being infected. I mean, along with the phage, there is only 1 phage genome inside. There is only 1 copy of C2, 1 gene copy of C2. Think of, and because there is only 1 copy, there will be certain degree of production, but there is also degradation. So C2 concentration will remain very low in this situation. And therefore, it will favor lytic cycle because C2 cannot activate the production of lambda repressor. Now think of in a different situation, MOI 3. MOI, multiplicity of infection. There are 3 phage genomes that are introduced into the cell. Now there are 3 copies of phage genome, C2, along with every other genes, of course. You have 3 copies of C2. That means you actually have higher concentration of C2 protein in this case compared with that of MOI1. Why is that? Here the copy number of C2 is high. That is because MOI is higher, right? So once you have a little bit higher concentration of C2, they have a possibility of C2 going and interacting with sorry, yeah, they have a probability that they will go and bind to the C2 binding site on one of these phages. And if it is binding in one of the phages, there will be production of C1 or lambda repressor. That lambda repressor will go and suppress the lytic genes from all these genomes. And at least one of them will express or will get integrated onto the bacterial genomes. Okay, I hope you understood that. I will try to quickly go describe this once more. So C2 is the master regulator of what happens, lytic or lysogenic choice. And that is dependent on C2 protein, and that is mostly about the stability of C2 protein, the concentration of C2 protein within the cell. So what we said was, if there are three copies of genomes integrated into the host cell because of multiplicity of infection, or there is only one copy, you will have C2 produced, but it will also be degraded by the host proteases like FTSZ. And if you have three copies, you will have C2 produced from three different genomes like this. And then that will build up a certain degree of concentration, because the number of proteases is constant, right? Proteases of the cell are constant. But you will have higher production in this case of C2 rather than compared to the degradation. In this case, in the MOI is 1. C2 is produced and the concentration or the activity of proteases within the cell is also high. So C2 is typically degraded and it will undergo lytic cycle. But when there is multiplicity of infection is high, C2 is concentration builds up a little bit because of three copies, and that will bind to one of the C2 binding sites like this case, and then it will activate the transcription from PRE, repressor establishment, which will allow the production of C1 or lambda repressor. Lambda repressor will bind to the operator regions that overlap with PR. Once it is binds with PR, it will block the transcription from Chrome. So it is acting as a repressor for this. At the same time, it is also activating the transcription from PRM. So in that sense, it is acting as an activator. Lambda repressor is a repressor of CRO gene, but it's an activator of lambda repressor itself, auto activator. You can call it as that way. I hope you understood that. There is one more protein that we haven't spoke about, that is C3. So C3, I think I have already discussed this part. Okay. So the stability of C3 is one of the points, right? C2 is very unstable because of a protein that is FTSH. I think I said FTSZ, that's wrong. That is encoded by HFL gene. C3 is a protein. C3 is a protein that stabilizes C2. How does it stabilize? Because it's an C3 is also similar in structure to that of C2. So it is kind of inhibits the protease activity of FTSH. And in that case, if FTSH is inhibited, then you will have the buildup of C2 protein concentration. I'll use some space here. All of it is dependent on C2. C2, the rate of production, and you will discuss this as rate of degradation. Degradation is brought about by FTSH, and rate of production is transcription from, you will have, say, C2 gene, I will put it as, and PR is the promoter. So if you increase the copy number of C2 gene by increasing the multiplicity of infection, then you will have more number of production of C2 while FTSH remains the same. There is another way of how one can increase the concentration of C2, that is by C3 protein. C3 protein also is degraded by or acted upon by FTSH. If there is higher production of C3, the degradation of C2 will be reduced, isn't it? If degradation of C2, because FTSH proteases, they are occupied with C3 protein. In that case, you will have the increased production of C2 or concentration buildup within the cell, and then that might also lead to lysogeny. Okay. So these are some things that you have to remember. I'll quickly review it. So CRO and C1. We discussed about CRO, which is important for, or which will allow the lytic cycle to proceed as such. We spoke about C1, that's the actual actor, I can say it as, that will impose lysogenation. There is another protein, C2. C2 dictates whether lysogeny should happen or not. How does it dictate? It dictates based on its stability, or I should actually say concentration. Any ways you take, you build up C2 concentration, then it is going to go for lysogeny. If the C2 concentration is low, then it will undergo lytic life cycle. And there is C3 protein also, which will increase the concentration of C2. So these three are working towards lysogeny. CRO is working towards lytic cycle. And we have already discussed about where the lambda repressor acts. It binds to OR1, OR2 with high efficiency. And if lambda 1 is produced, these two are occupied, OR1 and OR2. And there is also OL1, OL2, OL3. I didn't discuss those, but you can just, it's more complication than required. If lambda repressor is produced and it binds here, CRO is not produced, then it will undergo lysogeny. If CRO is produced first, it will bind preferably to OR3. That means C1 is not expressed, or lambda repressor is not expressed in those situations. That is because the operator regions are overlapping with the promoters. Okay. So if the first one, as soon as infection happens, PR is activated, and then as a result, CRO is produced, and C2 is also produced. C2, if produced in abundance, or the concentration of C2 builds up, then it will bind to C2 binding site and will activate the transcription from PRE, promoter establishment, or sorry, repressor, a promoter for repressor establishment, because of which lambda repressor is produced. Lambda repressor would bind to PR, something like this, because of which CRO is no longer produced, and there will be enhanced production because of activation of by lambda repressor, activation of PRM. PRM is promoter for repressor maintenance, and then there is more C1 that is produced. As long as C1 is high in concentration, it is lysogeny. And the production of lysogeny, or only in the decision making of lysogeny, you would whether it should be lysogeny or lytic, it is dependent on C2, right? And I will also, why they call it as clear, let me put it as C1, C2, and C3. I want to have a quick review in a different way. Let's try that. What do you think will happen if C3 is mutated? A mutation has happened in C3 protein, because of which it becomes non-functional or it is not produced at all. What will happen then? If C3 is not produced, it is going to decrease the stability of C2. How is that? By not engaging the FTSH. So C2 is degraded more. As a result, if C2 is degraded more, then you will not have the activation of C1. C1 repression is not established. Therefore, it will go lytic. When lysis happens, it will form clear plaques like this within plates. I'll try to find also um, the picture for it. Let's see if we can get. Yes, and 1 second. Yeah, I'll also come to this. Let's see here. So what will happen if C2 is mutated? If C2 is mutated or made non-functional, it's irrelevant what happens to C3 anymore. If C2 is not there, C1 is lambda repression does not occur, and then lytic phase is triggered. And also think of it like this. In the second scenario, if C1 is not there, then it's directly lytic phase is enhanced. There is no way scope for lysogeny to happen. So I'm trying to look for what is a clear plaques. I just found a page, and I'll probably be able to show you that. You see this picture here. Okay, I don't think I want to go there, but I just Googled it, and you can find it also. So here you see a clear plaque. That means all this thing that you're seeing here is actually bacterium. Let me see. Yes, this is a nutrient agar plate, and on that they grow bacterial cells, say something like E. coli, and they would have one spot or a little bit of phage that is made to infect. And here, whatever you're seeing here, the infection, and then you will have release of more phages, and those phages will infect adjacent ones, and slowly they will make a whole plaque like this. This is called as plaque, right? And that is clear plaque. There are also, this is not necessarily a translucent plaque. It is actually meaning, it could mean that the titer is less. You don't need to get into the whole details of it, but I think this is a good picture, hopefully. So, I mean, this is difficult to say, because what I meant to mean is there are different phages form different types of plaques. Some are very virulent, some are not so virulent, and so on. So it also depends on titer value and many other aspects. Trying to figure out where is a turbid plaques also will be there. Okay, I can't see it now, so we'll just let go of that. But I hope you understood what is a clear plaque. This is a clear plaque. If there is a non-functional or a mutation that will make C3, C2, or C1 non-functional, more probability of clear plaques forming and less of lysogeny. And that's why C stands for clear there. Okay, and I don't know if you can see, but this is a turbid plaque somewhere here. These are clear plaques. Okay, so you just remember these. These are why am I spending this much time on phages? All these are classical experiments, and if anybody is trying to go into core biology. Even if you're studying virology of viruses of animals or plants or anything, if we know the classic experiments, a classic understanding of this typical lambda phage is a model organism for a lot of viral studies, and that's how we know a lot more about it now, about phages or viruses in general. And that is why I'm trying to explain a little bit more in these details. Okay, I'll stop here. If you have any questions, I thought I will be able to finish a little more. I wanted to take up a little bit of translational regulation as well, which I will either post it as a video, or in the next